There is a pons, but it's the basilopontine nuclei only. You just can't use it, as George pointed out, as a topographic descriptor of the top half of the brainstem, in a sense. What Lewis has actually pointed out, and I didn't realise before at all, is that the pontine nuclei migrate down to the underneath of rhombomer 3 and 4, and that's where they sit. And although the pons is big in the human, it's really just like a huge balloon bulged out, and the stalk of the balloon is still in the same place, still in rhombomeres 3 and 4, but there's a big shelf on either side of it, which creates the confusion.
See, we are not identical twins.
Not at all. Doug?
In the spinal cord, you say they're actually more like 6 or 7 rhombomeres rather than...
Well, regions, yes, segments.
And are the boundaries of those regions coincident with the boundaries between what's always been thought of as the segments, segmental?
Well,
or do they kind of, are there boundaries somewhere in between?
No. The boundaries do fit in with the segmental plan, because the segmental influences are impressed overall, that apply to the vertebrae, do apply to some extent to the spinal cord. But basically the 6 regions are the 2 limb enlargements, and they have lateral motor columns, and then in between is the sympathetic preganglionic region, which is defined by HOX9, and then in front of the brachial area there's a C1 to 4 strip, which we call prebrachial, and then when you follow the lower limb subdivision, there's the parasympathetics fill in just for a couple of segments after that, and then the tail region. It is possible to divide the brachial enlargement region up into a top 2 segments and a bottom 3 segments on the basis of HOX5 and HOX8. But these are in a sense lineage restriction regions, which is what a segment ought to be. And I think, and I don't think this actually will upset people to see it like that, because if you want to see the evidence for it, you get a cholinesterase series of the spinal cord, and you see immediately where the change is. When you come from T1 to T2, you see the lateral motor column stops and the preganglionics start, and so they're quite sharp boundaries across those, but they do pretty well always occur at what would have been the traditional sort of vertebral segmental boundaries between those, if that explains it.
Well, we take all those considerations when we go for a boundary, how comfortable. We sometimes have the abbreviation BIIK, buggered if I know.
But it is true. I mean, if you look at the hypoglossal nucleus, you don't have any trouble. You can see the motor neurons there and all the markers, consistent things like that. But what we've generally done in the past is, where we really couldn't see a boundary, we just left it free. The problem is that doesn't suit 3-dimensional reconstruction. And what we're actually forced to do over the last couple of years was to go back and actually force those boundaries, even though we couldn't swear to them, because they're difficult in that way.
Nobody can sue us.
No, that's true. That's true. We can guess a little bit. I mean, I think the boundaries do work from individual to individual, but there's no magic in all of this. So when you're labelling 1,000 structures in 161 coronal sections, not everything has the same level of confidence as everything else, and some are very, very clear cut, and some are, I say the boundary should be here, George says it should be there. We argue for 10 minutes, say, okay, let's put it there, in fact, my wife was listening to us do this at one stage. She said, George and Charles, if anyone ever heard the discussion that goes on here, they'd never buy your atlas, because it seemed to be so arbitrary.
Yes.
She was making a point.
Yes.
In the introduction to the atlases, we do cover some of the more controversial areas to do with that. To be honest, we considered at one stage going right through every criterion that we would have for, say, why this is the red nucleus and why it's not the tegmentum or whatever. And it could be connectional, it could be histochemical, and so on like that. And we just realised it was going to be another lifetime's work. And to be honest, we backed off. And a more thorough scientist than we are would perhaps have done done that. Larry tried to do it to a significant extent in his forebrain stuff. He had the evidence for each one of those boundaries with a lot more references than we would have ever considered. But it is a lot of work, and so true, isn't it, George? Yeah. I mean, it's just you get defeated at times by the sheer volume of stuff. It's great to try and do an atlas of the whole of the brain, but you can't do everything for all of the brain. But it's a very good point. Where the delineation's been done on the basis of certain marker features, or there are other things of that kind, you could get guidance to do that. I mean, to be honest, we keep referring back to the chemoarchitectonic atlas of the rat brain, which has the 8 marker stains in sequence, because the signature of each nucleus can be projected onto other species from that. And we find that really important as a way of delineating, and I'd certainly recommend that people look at that kind of thing. So if you wanted to do a human area, what I would want to do is to see parvalbumin, calbindin, calretin, you know, a few of the good markers for that, and say if you're looking at the amygdala, and then you would be able to say, oh, I see, well, this is homologous with this because it has the same histochemical signature or you know, marker signature to do with it.
SMI32 for the cortex is the best delineator for primates.
Yeah, that's right. It doesn't work for everything, but you know, for example, if you're wondering about if you really can't recognise the inferior olive, then it is terrific in calbindin, and Purkinje cells are great in calbindin. So there are quite a few things you can say. It's characterised by the lack of staining in parvalbumin because everything around it is positive in parvalbumin, or something like that. In other words, just 1 or 2 sort of signal features which would be a quick cross-reference. It wouldn't be the same as a complete description, but it would give people a clue as to the way that you actually identified it. Whenever you're comparing different systems, you've got to have distinctive prefixes for them, and you're quite right. We're going to have to make the shift fairly quickly before we confuse people enormously. Thank you. That's a great idea.
One of the things that we're trying to do in BrainNavigator and that we're working on very heavily is to bring together into this one kind of structure-based environment different modalities of defining those structures. So be it by bringing in the chemoarchitectonic stains as an additional modality to show, or by linking out to the brain architecture management system that shows some connectivity data, and so on. So this is a real goal for BrainNavigator, is to really allow people to view directly structure-based information from different parts of neuroscience, and then bring that together and allow them to make their own decisions as well. So that's part of the goal for BrainNavigator.
Yeah, we haven't gone through that fine distinction of whether we're sure about this boundary, therefore we put a thick line, and this one a thin one, the other one a grey line, they go into the interrupted line.
We have considered that, I can tell you. But the point you're making is that you might end up with a couple of presumptive boundaries, both with good evidence for one or the other, and if there was a way of giving people a choice, that would be an interesting thing.
There is a molecular mechanism underneath the process of how these boundaries get formed. And I'd find it, so, and your description went into some detail there. Within the description of the ontology and Puelles' actual introduction and description of how this all fits together, how are those things described and how are they represented? Because I would imagine that would in fact be another interesting process. I mean, I would be very interested to see how one would represent that in a computational framework, how one would represent the whole process flow of the molecular events that give rise to these boundaries, in addition to the fact that there is now a boundary here. So is that something that is well described within the model?
It raises in my mind a really crucial thing, and that is that these boundaries are defined by developmental events, which say that a certain gene expression goes to here and it stops, and then something else starts on the other side of it, for example. The problem is relating those boundaries that are very clear in the embryo to what you actually have in the adult brain. Now, the availability of Cree lineages has made an enormous difference to that, although it still requires quite tricky interpretation, because what the Cree lineage can allow you to see see is that if HOXB1, for example, identifies the fourth rhombomer primarily in the embryo, and you have a Cree lineage, you can see exactly which things are labelled by that in the adult animal, so you can make the transition. But we can't do that for everything, and sometimes the gene is expressed in slightly different places at different stages during development, and so it's not a straightforward, linear transition. It requires careful interpretation, not all of which has been done at this point.
Is the data describing the actual molecular events available, and is it expressed as a knowledge representation? Is it expressed as at this point in time, this turns on in this location? And in fact, almost as a challenge to you guys as Atlas developers, what would be really cool is if you could actually show and illustrate which events happen at which time over the developmental period. I mean, I think that would be a terrific and important thing to see.
The answer is it doesn't exist.
We're looking for a good postdoc.
No, the answer is it doesn't exist in that form at the present time. But, I mean, the work of, say, John Rubinstein and Lewis and others that have defined many of these boundaries in the developmental series, that's all published and available and so on. But there's not what you're asking for, which is the natural extension of all of that. And that would be an exciting thing to be able to do, to be able to display those in that fashion. And I mean, the Allen Atlas series of developmental stages from E12 forwards that Lewis has done would be the basis on which you would do that. You would show from one stage to the other the appearance of gene markers, which then serve to separate this from that, or so on as you go forwards.
One thing that the Allen Institute is doing currently, in collaboration with Salvador Martinez in Alicante, is annotating the expression of 2,000 developmental genes that are available on our website using Luis's ontology
maps.
and his maps. And so
I hope the abbreviations too.
yes, of course. And so next October, so a lot of that molecular data about which genes are expressed at which stage of development according to this ontological system will be available publicly for people to analyze.
Great. I mean, Salvador was probably Luis's first doctoral graduate and has carried on the work. In fact, as Lewis points out, his lab is now 3 times as big as Lewis's is. He's a massive producer of data, and of course has worked particularly in the ISPIC organizer level. But he's a fantastic anatomist. He knows his chick anatomy really well.
Perhaps I should say that the existence of the Allen Institute has been a great bonus to neuroscientists, and that BrainNavigator would not have been here without the participation and the use of Brain Explorer from the Allen Institute.
Yeah, it's true. I mean, this isn't just forelock tugging. It's been such an incredible development, and the fact that it's been that it's preceded and now runs parallel to BrainNavigator just keeps enhancing both of the offerings for that. For example, in the new BrainNavigator, the Allen gene expression data can be pulled up alongside the Paxinos Franklin mouse atlas if you want to use that instead of the Allen atlas. You have a choice. You can look at both of them, and that's a fantastic thing. It just means that everyone can get the maximum out of the data that is available there on whatever kind of platform that they want to do it. It's a terrific thing.